So I just want to quickly say we're so delighted to have Dr. Sylvia Villeneuve here today. Dr. Villeneuve is an assistant professor at McGill University in the Department of Psychiatry. She's also an associate member of the Neurology and Neurosurgery Department. She holds the Canada Research Chair for Early Detection of Alzheimer's Disease. She's a member of the PREVENT-AD research program and an associate member of the McGill Center for Studies in Aging at the Douglas Research Center. And we're so delighted to have her here today to talk about her work predicting functional brain imaging in preclinical Alzheimer's disease.
Thanks a lot for the introduction. It's really a great pleasure to be here today, and I'm very glad to start this very exciting day, I have to say. So in my lab, what we do, I mean, it's quite an early lab. We started about 5 years ago, is that we're really interested into trying to identify individuals that will develop Alzheimer's disease dementia. So we know that the disease starts many, many years before people start to express these cognitive symptoms. And if we want to find ways to try to slow down or stop the disease, we need to be able to identify these individuals, both to put them into clinical trial, because we need to test medication on the one that will eventually develop Alzheimer's disease dementia. And then when we will have, hopefully, this medication one day, then to be able to give it to people, hopefully before they even start to have memory impairment. So that would be, that's kind of the general idea. So identifying these individuals that are at increased risk of developing Alzheimer's disease dementia. The talk will be divided in two main sections. I will present you actually two projects in which we use machine learning to answer clinical questions. And I will spend a little bit more time on the method of the second paper. So if there's things that are not, even if the methods are different, there's some redundancy there. So if there's things that are not completely clear in the first method, I will walk you through some of these steps again in the second project. So the first one was led by Jake Vogel. That is actually, so we met, Jake and I, when he was a research assistant at Berkeley University when I did my first postdoc. And he's now finishing a PhD with Alan Evan at McGill University. And the second project was led by one of my amazing postdocs that now left the lab to go back to Caen, where she's from. And in this second project, we developed a model to predict brain age based on functional brain feature. And then the idea was to see in the preclinical phase of the disease, so when people are cognitively normal, can we, is there any marker of the disease that accelerated brain aging? Always with this idea that, you know, what we want to do is that we want to try to identify individuals that will develop Alzheimer's disease dementia. And also, you know, if we could find marker that make people, let's say, age a little bit less fast, then maybe we would have better old age if we want so. Before I dive into these two projects, just that we are all on the same page for those of you that might be less familiar with Alzheimer's disease. So Alzheimer's disease is really a pathological clinical entity. And what that means is that you, of course, need to have the clinical symptoms, which are usually characterized by memory loss. But to know that it's really Alzheimer's disease in your brain, you also need to have the pathology. And here we're talking mainly about amyloid plaque and neurofibrillary tangles. And the hypothetical course of the disease is as follows. So we think that amyloid start in the brain of older adults and actually I say older adult, but this is false. I will tell you why in a second. But we think that amyloid start in the brain about 20 years before a person will get the diagnosis of Alzheimer's disease dementia. And then we think that amyloid leads to the spreading of tau, that tau is neurotoxic. This will impair the brain. And then at one point, people will start expressing mild cognitive impairment. And eventually, when they will not be able to do their activity of daily living, then it's when we will give in clinic the diagnosis of Alzheimer's dementia. Actually, we say Alzheimer's disease, but it's really when people meet the criteria, the more general criteria for dementia, really meaning that you're not autonomous anymore. And really what I want to stress out here is that there's many, many, many years happening before we give the diagnosis in clinic. So the good news is that we have many years to intervene on that disease. The last thing that I want to point your attention to is that there is two main forms of the disease, the sporadic one and then the autosomal dominant one. Sporadic Alzheimer's disease is what we're used to see in clinic. It's these older adults, usually they have 65 years and older, and there is no cause. We don't know the cause of sporadic Alzheimer's disease. We know that the main risk factor is age. Having a family history increases the risk of developing dementia by 2 to 3-fold. And being a carrier of the ApoE4 allele also increase your risk of developing Alzheimer's disease. The ApoE4 allele is not causing Alzheimer's disease, but it's working on different pathways, such as a cholesterol transporter. It also helps for the clearance of amyloid. So this gene seems to, if you are an ApoE4 carrier, it will increase your risk of developing Alzheimer's disease. But again, it's not causal. The other form that people know less about, it's less than 1%, it's autosomal dominant Alzheimer's disease, or AD. And this form of the disease is really interesting because we know the cause. So this disease is caused by few genetic mutation, and then when you heritate the genetic mutation from your parent, you will for sure develop the disease. And you usually develop it within your 30, 40, or 50, so way earlier than for sporadic Alzheimer's disease. In the talk, I will talk about many cohorts, but the two main ones are the PREVENT-AD cohort and then the DIAN cohort. The PREVENT-AD cohort is a cohort that is based at McGill University. We are following about 350 participants that are cognitively normal at entry. We're cognitively normal. We have less than 10% that develop cognitive impairment or dementia throughout the longitudinal follow-up. We started the cohort in 2011. Actually, John Breitner started the cohort. I was not yet at McGill University. And then, sorry about that. And then the NL, so this is really, and so we're really targeting individuals that we believe are at increased risk of progressing to Alzheimer's disease dementia. The other cohort is the DIAN cohort. It's a cohort following individuals with AD. The main core of the cohort is based at Washington University, but given the rarity of these individuals, there are many sites recruiting or testing these individuals. One of them is at McGill University also. One of the most famous paper related to autosomal dominant Alzheimer's disease. And this is important because this years to estimated symptom onset that I will describe in 1 second is what we try to predict in our first predictive model that was conducted by Jake Vogel. And so in individual autosomal dominant Alzheimer's disease, as I told you, if you have the genetic mutation, you will develop the disease. And then we know about when you will develop the disease. So you should develop the disease about at the age at which your parent developed the disease. So let's say that your mother had the genetic mutation. She developed the disease when she was 30 years old, and you are 20 years old, then you are 10 years away to develop the disease. So on that graph, if you look at the x-axis, you would be -10, and so on. And then so they use in that paper cross-sectional data to put every individual on this scale of how far they are from the age of onset of their parent, or what we call years to estimated symptom onset. And what they did is that they looked at the different trajectory of the different biomarker. So every line here represents a biomarker of Alzheimer's disease. So for example, we have cerebrospinal fluid, amyloid beta, the yellow line, that we think is about one of the markers that we can capture first as being abnormal within these individuals. And we think that this biomarker is abnormal about 20 years before their expected symptom onset. Again, we would love to do that using predictive model and then using longitudinal data, but these data don't exist yet. We're working on acquiring them. So one of the first things that we did when I arrived at McGill University looking at the PREVENT-AD cohort is that we asked the question, well, is this a relationship between the age of the person and then the age of the parent that here we call years to estimated symptom onset? Is this related to any biomarker in the PREVENT-AD cohort? And then what we found is a weak but significant relationship between estimated years of onset and abnormal level of amyloid within the CSF. Cerebrospinal fluid. when we saw that, we said, well, it's not that strong, so let's ask for other cohort. We replicated that finding in other small samples, and a few years later, now we have PET imaging in a subset of that cohort. We replicated the finding again using PET. But, the association is there, but, is it strong enough to say that if you have someone in front of you in clinic, you will use that score to try to, predict how abnormal the amyloid is in the brain of that person? Looking at that graph, well, I don't think so. I wouldn't trust doing that. So we also found association between this estimated years to symptom onset and some functional and brain feature. And then what we decided to do, because we had more participants, is that we decided to try to create a predictive model to see can we actually create, let's say, a brain signature and then predict this estimated years of symptom onset based really on functional resting state data and then structural data. And then to walk you quickly through the processing method, what we did is that we took all the resting state data, pre-processed them using NIAC, then segmented the brain in 443 cortical regions, from which we extracted time series in each of these regions, and then we created a correlation matrix for every single participant. For the structural MRI, so we segmented the brain using SPM12 and then just processed them using kind of a regular VBM pipeline in order to get probability map. And then really the outcome here is really that we, based on that, we extracted gray matter volume in these 443 cortical region. And the important part here is that what we did is that we use these now 887, because we include also the total intracranial volume, these 887 brain feature as our predictor in our model. So we build a predictive model based on these functional and then these structural brain feature. Out of about 250 participants, we had 206 participants that passed all the QC for both modalities. We were not dealing with missing data here. And then what we did is that we split our sample into a training set to build a model and then into a test set. So as for the training sets, we did a threefold cross-validation approach. So, and really what is important here is that, you take all your data and then you decide to really to build your model just in part of your data set. So this is this training set here. And then the technique that we use to build our predictive model is a lasso regression. And then at the end of the day, so we build 3 different models based on our training set data. And then what we did for our final model is that we averaged the weight of all the 3 fold or the 3 model. And this is what we apply to the unseen data. And one thing that I didn't mention here, but another step that we did is that in the final model, we're really only keeping the feature that were significantly associated with years to estimated symptom onset, trying to have a model that is as simple as possible. Now to the results. So again, we did that in 2/3 of the cohort, and then for the 1/3 that we didn't touch. So now I'm presenting you when we are testing the model. So what you see here, I guess that the axis should have been changed, but so what you're seeing here on the x-axis is the predicted years of onset, and on the y-axis are the real data, so the real years to estimated onset. And when I say the real data, again, it's just this calculation of the age of the parent. So actually the age of the person minus the age of the parent. So this is what we're talking about here. And this model, even if not perfect, because if you look at the correspondence between the number themselves, it's a little bit off, but still we're explaining 22% of the variance. And then in general, what we can conclude from that is that the more you express this brain signature that I'm showing here, then the closer you are to the age of onset of your parents. And when we look at the feature that were included in the final model, well, we were reassured because for those of us working with Alzheimer's disease, these brain regions are often found to be impaired early in the course of the disease. So if we look, for example, at the resting state functional feature that were kept in the model? Well, I mean, we have brain regions such as the hippocampus, the angular gyrus, the precuneus, so all brain regions that are extremely vulnerable at early stages in the course of the disease. And then, of course, the real question is, well, it's good, like we use these fancy techniques, but is that better than what we have in clinic? So what do we have in clinic to try to predict who will progress to Alzheimer's disease dementia? Well, often we have demographic information. So in this case, we're talking about maybe preclinical sporadic Alzheimer's disease, so the age of the person. We have family history in this case. We have cognitive score. Here to compute kind of a clinical model, we also use APOE4 information, which is not always available in clinic, and then the hippocampal volume. And then the idea of this figure that I'm showing you is to ask the question, was our predictive model better at predicting years to estimated onset than another model that would have been built using a similar technique, but then in this case would have been built only using the clinical information that we have in clinic? And the answer is yes. So our model will be here model 1, and then the clinical model will be represented as model 2, and you can see that our model outperformed the clinical model. Now, if we put both information together, which is a little bit intuitively what I would have done as a clinician, then the variance is a little bit better. So this is model 3, but it's not statistically better than the brain signature that we developed, so model 1. And then, of course, like you might think, well, this is great, but this is kind of, at the end of the day, you're just predicting how far you are from the age of onset of your parent. So Jake, looked very hard in the field for data, longitudinal data, in which some individual would have progressed to mild cognitive impairment or dementia throughout the course of the disease. These data don't really exist. There are only few of them. And then he found 26 individuals from the ADNI cohort. This is an open access cohort in which we had these individuals that through the course of the longitudinal follow-up, they started by being cognitively normal and then convert to have cognitive impairment. And what we're seeing here is that if we apply our predictive model, these brain feature, then we're able to predict how far people are from the progression. And actually, what I just said is a little bit wrong. We should maybe more rephrase it, because of course there's some error in that model, in the way that the more you express this brain signature, if you want to, then the closer you are to really disease progression. So to conclude for this first part of the talk, so I show you that, so in individual with autosomal dominant Alzheimer's disease, we know when people should develop the disease, because that should be about at the age of onset of their parent. The first main finding that we found in the PREVENT-AD was really that this association might also be present in sporadic Alzheimer's disease. And this is kind of a big finding, because not everyone in the PREVENT-AD will progress to Alzheimer's dementia. So even though if this relationship is far from being perfect, it's still quite strong, because while having some non-progressors, some individuals that are not in the preclinical phase of the disease in the PREVENT-AD, we're adding really a lot of noise to our data set. So if we find this relationship, and then we found it using correlation with amyloid in different sample, and then using predictive model, looking at structural and then functional brain feature, and probably even more importantly, we're using a completely different data set, and then asking a little bit a more different research question, which is really eventually what we're trying to get at: who will convert and when, then this model was working. Now, before I conclude really on this first section, I just want to do a little plug-in. We are working very hard with the Canadian Open Platform Initiative, so Julie Pauline, Alan Evan, to put the PREVENT-AD data open access. It is a long process. It took about a year just to reconsent most of the participants. If you look at the openpreventad.loras.ca website, you can find MRI and sonographic information on about 200 participants. We are working on putting more data available, so more participants, and also more more information there, because right now it's just the demographic, and eventually soon, hopefully, the amyloid PET and tau data on a subsample will also be put there with some MIC data. Okay, so second project. So all the project that we do in the lab, we do it really in a collaborative way, because in the lab we have individuals that have a lot of skills in method, and then we also have individuals that are more clinician, for instance. So so when I say in the lab, it's the lab and then outside of the lab, because one person that advised us on the two projects on the method part is Etienne Vachon-Presso, and he's actually a faculty at McGill University as well. So in this second project, the idea was really to build a model of brain age that worked across lifespans. So that was the first challenge, and based on functional brain feature. And why functional brain feature? Well, it's because we think that we might be able, in the preclinical phase of the disease, to capture functional change before structural change. And then the second part of that project was really to test, is amyloid and then some genetic variant related to accelerated brain aging. And this project was led by Julie Gonod, again, so a previous postdoc in the lab. This idea of building a model of brain age is something that I wanted to do for many, many years, with this idea that I've always been intrigued by the fact that not everyone seems to age at the same pace. So it's very impressive when you have someone in clinic in front of you that is 7 years old, but look physically, but also cognitively, just like if they were like 50 years old. Well, just after that person, you have another person with the same age, and then this new person look way, 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 way older. And often when you do the cognitive testing, this person look on the test is way worse as well. And in fact, there have been some twin study where they looked at twins, and then they asked people to rate the age of the person based on the wrinkles and things like that. And then the twins that was looking older often had more cognitive impairment. And in the longitudinal study, the twin that was looking older was dying, had higher chances to die in the course of the longitudinal study. So really suggesting that, we have our biological age, we have our chronological age, so when we were born, but this biological aging, how fast do our organ age, this seems to be important in order to live well longer. To give you an example, here are two brain of individuals from the PREVENT-AD study that have about the same age. Both are cognitively normal. And then what you can see is that the person on the upper part have a brain that look way younger than the person on the lower part. On the lower part, there is a lot of atrophy. For those of you who are not used to read these scans, if you look just at the size of the ventricle, the ventricle are really way larger. And then there's also more atrophy within the temporal lobe. And so in order to target this question of chronological versus biological aging, few labs, or actually more and more labs in the world, have developed model of brain age. And then these predictive models usually use structural features. In our project I'm going to present, we decided to go with functional feature again, because we think that they might be earlier marker of Alzheimer's disease. And what the literature is suggesting is that if you have Alzheimer's dementia, mild cognitive impairment, or other condition, often you will show accelerated brain aging. Here is one study that I want to give you a little bit more example about. I think it's very interesting for the topic of the talk. So if you look at the panel on the left, the groups are split into cognitively normal individual with mild cognitive impairment, that are again split in two based on who were stayed stable throughout the course of the study. It was a longitudinal study, and then who through the course of the study progress to Alzheimer's dementia. And then the final group is this group of individuals with Alzheimer's dementia. And then what they're showing here is that even at baseline, when everyone in the group of stable or progressive MCI were considered as having mild cognitive impairment, the one that eventually progressed to Alzheimer's dementia were already showing accelerated brain aging when compared to stable MCI. So suggesting that maybe this accelerated brain aging is a biomarker of disease progression. And what you're showing on the right, what what I'm showing here on the right is a paper from the same group that worked a lot on that topic in the field of Alzheimer's disease. And then again, they are showing that individual with mild cognitive impairment, so the progressor Alzheimer's dementia, are showing accelerated brain aging over time. And then what this figure is adding is that if you are a carrier of the APOE4 allele, so again, this genetic risk factor for the sporadic form of the disease, then your brain is even aging even faster. So it's kind of a two-hit effect. So you have the disease that make your brain age faster over time, and then having this APOE4 allele also accelerate this brain aging. Now, really the research question that we'll target in a second is, well, what about if you look at cognitively normal individual? Can we see accelerated brain aging related to Alzheimer's disease risk or causal factor? And, that question might look trivial because you think, oh, well, they already did it in the NCI and then so on, and then why always targeting to these little individuals? Well, there is about 1/3 of older adults that are aged 65 years and older that are walking down the street with a brain full of amyloid. So, targeting really these individuals and then trying to stop the disease is probably not that trivial. So for the first part that I will show you is really how did we develop the model. And again, it's important to stress the fact that we wanted a model that spanned across life, so not just an older adult, which would probably have been easier to do. And this idea is because we want to also test some association within the DIAN study. And as I told you before, these individuals are way younger. So we add, after QC and things like that, we grab data from many different data sets. And then for the project, the final data set on which we created and tested the model was 1,340 individuals. And then what we did in the first step is that we split the data into 3 groups. So the training set was really used to build the model. And then after that, to make sure that we were not overfitting the model, we used the validation set. And I will walk you through that in more detail in a second. And then only when the model was final, we passed actually months developing the model. And only when the model was final, we, what we said, we called open the envelope and then test if our model was working in the test set, but also test our research question within the test set. And we never tweaked back the test set. We just worked with what we had, and this is what will be eventually published. It's in preprint right now. To walk you quickly through the method, so the scan again, so now we only have resting state fMRI in our predictive model. They were processed again using YAC. We use a different parcellation, so we use the Power and Peterson Atlas, and we extracted time series within each of these parcels. Now we're talking about 239 nodes. Created again correlation matrices for every participant. And from these correlation matrices, we computed using the Brain Connectivity Toolbox 26 graph metrics. And then before developing our neural network, we did a first step in which we looked at actually all of the 26 graph metrics and tried to predict using a support vector machine and then also regression tree assemble, which of these features might actually be the best predictor of chronological age. And I will spend a little bit more time on that within the next slide. But the idea was just to have a general idea in order to rank the feature by whom would be the best predictor, to feed them first in the model. And then after that, we use MATLAB to build the neural network. And here, how to read the model, it's really that the input are the 26 graph metrics. So here in this example, we are putting the five graph metrics in the model. And then the output is really this chronological age that we're trying to predict. And then, so I mean, this was not done with Nilearn, but there's many steps that you can do using Nilearn function that are here in orange. Okay, so if we spend a little bit more time on the architecture of that network, so here would be an example of the simplest model that we created. So 5 feature, again, these are 5 graph theory metrics, and then with one hidden layer that is composed of 5 nodes. And again, we want to try to predict the output, that is chronological age. And the way we decided in that study to assess the validity of the model was by looking at the root mean square error. And so the more error, the worse is the model. So if you do a very, very simple model, I mean, it kind of makes sense. I mean, you will add a little bit more error. Now, the best model for this in our training set would have been a very complex model. And again, it kind of makes sense because the more precise the model, the more complex the model, the better it will be at, predicting the age in your participant in which you're building the model. However, the problem with that, if you take this very specified model to your training set, is that it's not going to be generalizable to another data set. So it's what we call overfitting. So it's why here we took a different data set. It could have been a subset of the different data set, but here we really decided to go with a completely independent data set that was also, that had individual crossing the lifespan. And then what we did is that we test actually the generalizability of our training set within this new data set. And again, what you can see here is that if your model is too complex, it's just not going to work. So to define our final model, and then this is the model that will be used in the next step when we will test more the clinical question. So we decided to go with the model that was the best generalizable. And this would be, in this case, so 10 feature and then 2 hidden layer. And I told you in the previous slide that we did this feature ranking first. Well, the idea was really that if you start by feeding the model with the feature that have the most chances to predict age, you will have a more simpler model. If you don't do that and then you just enter the feature randomly, your model will be way more complex and probably it's going to be more difficult to generalize it. So we think that this first tweak was probably a good implementation. Now to the results, I mean, in the training set, so I mean, this is, I guess, less exciting because we use this training set to train the model, so of course it should work. So what we're seeing is that we're explaining 53% of the variance. What is probably more impressive is that the mean root square error is quite low. At least we were very happy about that. And again, it's a model that is spanning across lifespan, so not that easy to implement. If we look at the validation set, so unseen data that were not used to train the model, again, it's working extremely well, explaining about 50% of the variance, and again, very little, according to my understanding, no error. And then now when we open the envelope, so again, like this is completely unseen data, and then we're not deciding how generalizable is that model. It's really kind of, we are looking at that model for the first time. And we did that again, like a few months after we started the project when everything was done, but we were quite happy about the results. So we're explaining 36% of the variance. Of course, you can see that some data sets, just as the PREVENT-AD, are a little bit overestimated in terms of age, but overall, for a model on which we never tweaked based on previous data, we were quite happy. And to the last part of this talk, so the last question was really to test if this predicted age model differences, so difference between predicted brain age and then chronological age, was associated with genetic factor or amyloid factor. These are the characteristics of the individual of interest that were included in the test set. And of course, the main difference between both cohorts is that the PREVENT-AD participants are way older by definition because they're at risk of sporadic Alzheimer's disease. In order to test our hypothesis, what we did is that for individuals in the DIAN cohort, we split the group as based on the one that do have a genetic mutation versus the one that don't have a genetic mutation. And then in the PREVENT-AD, we split the group based on the one that have 1 or 2 APOE4 allele versus the one that didn't have APOE4 allele. So yeah, they were APOE3/3, for example, or 3/2 or so on. And for the amyloid, so we pre-process the scan using a quite standard method where we use an MRI that we co-register to the PET scan in order to define some region of interest. And then we extract standardized uptake values ratio within a lot of brain region because we know that when amyloid is there in the brain, it's pretty much distributed in a lot of brain region, and we use the whole cerebellum as the reference region, because if you just look at kind of the raw PET binding data, well, you will have the tracer will bind to things that will not be amyloid. So usually we normalize by a reference region to really try to extract only or mostly the signal related to amyloid in the brain. And we use the whole cerebellum to do that, because it's a brain region where usually there is no amyloid there, at least not in the first stage of the disease. And we add different tracer within our two cohort. And, you know, like 10 years ago, that for me would have been a big limitation, and I would have spent time in the paper discussing about that, you know. And then now the way that I see that is that if you can build a predictive model and then it works, you know, when changing the data set, when changing the tracer, when changing sometimes a little bit the region that you're targeting and things like that, it means that it's probably generalizable. And if you want to try to build a model that you can import in the clinic, you need to have something that is a little bit messy, because clinic is not messy, but it's you know, it's challenging to predict something in real life. So for me, you know, now dealing with the cohort that have different tracer and things like that, it's a good thing and then not a bad thing. So the mentality is changing in the field. Now going to the results. So in the DIAN study, so we found that individual with a mutation carrier, so individual that we know will develop Alzheimer's disease, dementia. And in that study, again, we only included cognitively normal older adults. So their brain was predicted as being older when compared to non-mutation carrier. In the PREVENT-AD cohort, we didn't found an effect of ApoE4 on this predicted age signature or model. And when we saw these results, I mean, of course, I was 100% sure that this finding in the mutation carrier was influenced by the fact that some of them already have amyloid in their brain. You know, we think that amyloid is the first marker starting in the course of the disease. So I was really sure that amyloid was driving this effect. And I was extremely surprised when I saw that it was not the case. So here, when we split the group of mutation carrier based on the one that don't have amyloid and then the one that have amyloid, the one that don't have amyloid, that is in the middle here in the lower panel, they also have this accelerated, brain aging. So suggesting that it's not necessarily amyloid that is causing that finding. And then again, in the PREVENT-AD, no association with amyloid. And then because we were quite surprised about that finding, we decided to look at amyloid as a continuous variable. So looking at an association between this predicted age difference and amyloid as a continuous variable, really asking the question, well, maybe, I don't know, like the one with high level of amyloid will drive this association. And again, it was not the case. So to conclude on this last part of the talk, so we were able to create a model of brain age based on functional feature. The individual with autosomal dominant Alzheimer's disease that were mutation carrier, they had accelerated brain aging compared to the non-mutation carrier. And if you're asking your question, who are the non-mutation carrier? Well, it's just that when you enter that study, you need to have a parent with autosomal dominant Alzheimer's disease. If you heritate from the gene, you're in the mutation carrier group. If you don't heritate from the gene, then for sure you're not in the preclinical phase of Alzheimer's disease because you're too young. So you're in the non-mutation carrier group. So a perfect group control, actually. And then this association was not accounted for amyloid in the brain of the mutation carrier. If we go back to this hypothetical model, I mean, it challenged it. And what it suggests is that maybe actually, in fact, differences or change in resting state functional connectivity happier prior to amyloid deposition. And if you want to know or read more about that topic, there is a nice review by Bill Jagis and Beth Marmino on that topic, where they talk about a lot of, actually not a lot, but few studies that have done in rodent, suggesting that the abnormal brain activity might actually lead to the accumulation of amyloid. And as a General conclusions. So so predictive model can be used to answer a clinical question. And my best advice for those of you clinicians that want to do that is start with your question. Don't try to build a predictive model. Just start with a clinical question. And then after, instead of doing a simple regression or like a correlation, ask the question, can I answer this question? Do I have enough subject to try to build a model and then see if I can, predict my outcome? So I think this is probably the best way and the simplest way to start. In order to build these predictive models, you need a lot of data to train the model, and then you also need an independent test set to test your model and be sure that your model is generalizable. And really, this is what we want eventually in clinic. Given the increased number of data sets that are open in the field of Alzheimer's disease, building predictive model in Alzheimer's disease is more and more feasible. However, if you want to do that, it's still extremely challenging because Alzheimer's disease is an heterogeneous disease by nature, which is fun to build predictive model, but that also means that you need a ton of data to have a model that will really fit the heterogeneity of what you're trying to target as an outcome. And then the other thing is that reviewer will often ask you to kind of, review logical data, for example, or something like that to validate your finding. And as for now, these data don't really exist. So it's really the main challenge that we have at the moment while building model on Alzheimer's disease. So with that, I thank you all for your attention. A special thank to Jake, Julie, that led the first project, Etienne that helped us with all the analysis, Alex as well that really built the brain age predictive model, and Alexa that helped throughout all these different projects and actually all the projects in the lab. Thank you.
Thank you so much. That was a really lovely talk. Okay, so I think we have time for maybe one question. There are 2 questions in the Ask a Questions tab. So either if you'd like, maybe you could answer one or you could try and address both quickly. If it's easy, I can read out maybe what the questions are.
Read them and then, yeah. And then if you have a question, just please send me an email and I'll answer them.
Perfect. Thank you. Okay, so maybe the first question then would be, have you considered how the correlation with brain signatures may differ pre and post Alzheimer's disease onset?
Sorry, say it again. So if I was trying to read them, but I cannot find them. I'm missing now.
No, it's fine. So the question was, have you considered how the correlation with brain signatures may differ pre and post Alzheimer's disease onset?
Yeah, absolutely. So what happened with Alzheimer's disease onset is that, of course, the brain signature would be way more important. But I think that at one point, because all the brain is so destroyed, I think, there would be kind of this curve where at first the brain signature is hard to implement, it's hard to find it, right? And then after that, the more the disease progressed and the more people will express it. And then at one point, after the diagnosis of Alzheimer's disease, dementia, my guess is that the brain is kind of so atrophied at that point, so messy, that maybe then it's kind of harder again to find this brain signature. But this is just, a thought, but something to be tested for sure.
No, I think that's a promising perhaps area for sure to think about. Okay, I just want to say there are a few more questions, but I don't think we'll have time to get to them. I will make sure they get to you. But thank you so much again. This was a really lovely talk and I really appreciated it.
Thank you.
All right, so I will close this broadcast and I will see everyone in the next session. Thank you so much.
Thanks.